Hello friends, hope you all are well and doing good. Welcome back to my channel. In my previous video, I talked about construction of first artificial recombinant DNA, and today I am going to take tools for recombinant DNA technology. It is just like that. If you need to win a war, you need good weapons, and for a technology to be successful, you need tools. Here in the recombinant DNA technology, the tools are your enzymes like nucleases and cloning vectors. For better understanding of the topic, you should familiar with the terms like DNA, RNA, nucleotide, nucleoside. What is the difference between all these terms and phosphodiester bond? So let's have a brief account on all these terms. DNA, RNA, and in some cases proteins are the genetic material. DNA is the name given to the chemical that carries genetic information in all living organism. It consists of two strands that wound around each other to form a shape or structure known as helix. Here you can see in the diagram there are two strands, one in 5' to 3' direction, the other one in 3' to 5' direction. Both these strands have sugar phosphate backbone, and to each sugar a nitrogenous base is attached. In case of DNA, the four nitrogenous bases are adenine, guanine, thymine and cytosine, where adenine and guanine are purine, whereas thymine and cytosine are pyrimidine. The sugar in case of DNA is deoxyribose sugar. RNA is very similar in structure to that of the DNA but differ in subtle ways. Here in RNA, the sugar is ribose sugar. RNA is single stranded, whereas DNA is double stranded. The four nitrogenous bases in RNA are adenine, uracil, cytosine and guanine. Often students get confused with nucleotide and nucleoside terms, so let's have an understanding on these terms. Nucleoside is made up of nitrogenous bases, usually either purine or pyrimidine, and 5-carbon sugar. Nucleotide simply a nucleoside with additional phosphate group. Phosphodiester bond. What is the phosphodiester bond? It is the linkage between the 5' carbon of one sugar and 3' carbon of the another sugar. Here you can see in the diagram, this is the nitrogenous base thymine, this is a ribose sugar, this is the 5' carbon of the ribose sugar, and here is the 3' carbon of the ribose sugar. Again, this is the nitrogenous base adenine. This is a ribose sugar, and this is the 5' carbon of the ribose sugar. Here between the 5' carbon and 3' carbon of two ribose sugar, there is a phosphate group which forms a phosphodiester bond. Enzymes play a vital role in all biological processes. Here the first tool for recombinant DNA technology are the enzymes nucleases. These enzymes act by cleaving the phosphodiester bond between nucleotide subunits of nucleic acid. Here you can see that nucleases are the enzymes that are capable of catalyzing hydrolysis of nucleic acid by cleaving phosphodiester bond between nucleotide subunits of nucleic acids. Nucleases are of two types, exonuclease and endonuclease. Let's have a look. Nucleases are of two types, endonuclease and exonucleases. Exonuclease act by removing the nucleotide from the end of the DNA, whereas endonuclease act by making a cut at the specific position between the DNA. Restriction endonuclease, or you can say restriction enzyme or molecular seizure, they come under the category of endonucleases. With this, I am ending this video now. Hope these videos might be useful for you people. In my upcoming videos, I am going to give a detailed description about the restriction endonucleases, how they act, how they are named, what is the difference between a sticky end and blunt end. So please subscribe it. One more thing, thank you for all your love and support to my channel. The viewers who have viewed my channel but not yet subscribed it, please do subscribe it. Till then, good day, bye bye.